Today I'm going to talk about the innate immunity. It is non-specific, so it is not specific against each and every organism. For example, if you think of bacteria, they are classified into gram-negative, gram-positive, mycobacterium, spirochetes. Similarly, viruses, RNA, viruses, DNA, single-stranded, double-stranded. They will take pathogens as groups. There are three lines of defense. The first line of defense in our immune system is barriers, and I'll talk about all these one by one. And then the second line of defense is cells of innate system and humoral component. By humoral component, I mean complement proteins, C-reactive protein, things which are soluble in the blood apart from antibodies. So antibodies are not included in this. The second line of defense include innate cells and CRP or complement proteins. And the third line of defense is B lymphocytes, T lymphocytes, and of course plasma cell and antibodies. So that's the third line of defense, the adaptive system. So you think below this line is the adaptive immune system. Above this line, the first two barriers, they are part of the innate immune system. Now let's start with the barriers first. So this you can see is the epithelium. This can be epithelium of the respiratory tract, of the GI tract, or it can be simply epithelium of the epidermis of the skin. So this is going to act as the physical barrier. The first is acting as a physical barrier. There are chemical barriers like alpha defensin, beta defensin. They can kill gram positives. This can kill gram negatives. Viruses, cathelicidin can kill mycobacterium. Lysozymes is present in the tears. It can affect a lot of pathogens, and everyone knows the acidic environment in the stomach. It is also very helpful against pathogens. The third component in the barriers is the mechanical. So on the certain mucosas, there are cilias on the surface, and these cilias are also very helpful, and they move the pathogens in the respiratory tract. So that is the mechanical barrier. Finally, the fourth component is the biological. And biological, everyone know the normal commensals that are present on the skin in the GI tract, and there are around 500 strains of the normal commensals that we know. As the second line of defense, we have got dendritic cells in the tissue. So immediately below the skin or the mucous membrane, we have dendritic cells, and also are macrophages. You can see that there are macrophages, a dendritic cell, and this is a mast cell. First thing, the dendritic cell that is present in the tissues is called as immature dendritic cell, and I'll talk about that why it is called as immature and how it becomes mature. The macrophage that is present in the tissue, that is called as resting macrophage. And the mast cell, it has got in its granules lot of potential chemicals and mediators, but it is not yet degranulated. So these three cells which are present in the tissues all the time, these cells are main cells of the innate immune system. Here NK cells, natural killer cells, even though they come from the lymphoid lineage, but they function with the innate immune cells. Neutrophils are present there as well. These monocytes are the cells which, when later comes into the tissue, they will turn into macrophages. That is called as basophil. They can release histamine, prostaglandins, leukotrienes. So they are the equivalent of mast cells in the blood. The eosinophil. And apart from this, the soluble substances that are present in the blood, they include the complement proteins. So we know that complement proteins C1 to C9 and then B, D factors, all they are present in the blood as inactive form. So, but they help as innate component is IgM antibody, the primary antibody. But the main cells and the humoral component that you need to remember is dendritic cells, macrophages, and mast cells in the tissues, and complement proteins, reactive protein, natural killer cell, neutrophils, monocytes, basophils, and eosinophils. They are present in the blood vessel. So these are the receptors. They will detect, TLR2 will detect gram positive pathogen, TLR4 will detect gram negative, TLR3, 7, and 9 will detect viruses. Viruses are all obligate intracellular. So this virus entered the cell. Inside the cell is receptor TLR3, TLR7, TLR9. They will detect this virus. When they detect this virus, these epithelial cells will now produce interferon alpha and interferon beta, which will create antiviral state. Now these dendritic cells also have toll-like receptors like macrophages, and they also have some other receptors which are very important, called as complement receptor. Another one is called as Fc gamma receptor. And so let's bacteria which has entered here in the tissue. The complement protein, they are activated by three different pathways: the classical pathway, the mannose binding lectin pathway, and the alternative pathway, and they produces C3A, C4A, C3B. I will just write the important ones. This is called as MAC formation. Now C3A, C4A, C5A, these are anaphylatoxins, while C3B, C4B are opsonins. These opsonins will coat the bacteria. And when they coat the bacteria, this complement receptor which is present on the dendritic cell, or the one which is present on the macrophages, CR, complement receptor, this will recognize this opsonin. When it is recognized, it will lead to phagocytosis. That is number one. Number two, I told you these antibodies are produced IgM and IgG. IgG, when they come out of the bloodstream, they are also able to coat these bacteria. And when they coat the bacteria, there is a receptor for these antibodies as well, and that is called as FC. FC is from this FC part of the antibody. Gamma R1 is because of it's against the gamma IgG, so that's why it is called as FC gamma receptor. So it's the complement receptor, FC receptor, toll-like receptor. They will all help in one thing. They will act as opsonins, and they will help in the process of phagocytosis. The classical macrophage's job is phagocytosis and killing, while the alternative macrophage M2 job is tissue repair and regulation. So when the job is done, the fight is over, these M2 will be activated and they will help in tissue repair and regulation. And the main cytokine that is involved in alternative macrophages is IL-4. Whereas the classical activation and M1 reaches its maximum activity with the help of interferon gamma. These are produced by a lot of cells, like for example, NK cells can produce interferon gamma, T lymphocytes can produce interferon gamma, and IL-4 is produced mainly by Th2 cells. So this, at the moment, what you need to remember is that the macrophages, they are mainly of two types, M1 type and M2. M1 helps in phagocytosis and killing, M2 helps in tissue repair and regulation.